Plant breeding is the skillful use of different plant varieties to produce a desired plant type, thereby improving crop yield. But in most cases, crop production is affected by several crop diseases caused by pathogens such as fungi, bacteria, and viruses. Blast of rice, red rot of sugarcane, and late blight of potato are examples of the fungal plant diseases, while black rot of crucifers is a bacterial disease and tobacco mosaic virus and turnip mosaic virus are viral diseases. In such cases, plant breeding can be used to develop crop processing resistance to a particular pathogen, provided the causative pathogen and its mode of transmission is known. Resistance can be defined as the crop's ability to prevent a pathogen from attacking it and is determined by the genetic makeup of a particular crop. The genetic makeup of a crop can be altered using plant breeding techniques like conventional breeding and mutational breeding. Conventional breeding involves hybridization and artificial selection. It involves several steps that include collection of germplasm, selection of disease resistant parents from germplasm, hybridization of the disease resistant parents, selection of disease resistant hybrids, and evaluation of the selected hybrids. There are many disease resistant hybrid varieties of different crops developed through conventional breeding. Let's take a look at some of them. Himgiri, a hybrid variety of wheat, is resistant to fungal diseases such as leaf rust, stripe rust, and hill bend. While Pusha shubra, a hybrid variety of cauliflower, is resistant to fungal diseases such as black rot and curl. There is Pusa komal, a hybrid variety of cowpea that is resistant to blight diseases caused by a bacterium, while Pusa sadabahar, hybrid variety of chili, is resistant to viral diseases such as tobacco mosaic virus and leaf curl. The only drawback of the disease resistant wild relatives is that they are low yielding. So, the disease resistant genes from these wild relatives need to be transferred to high yielding cultivars. As the disease resistant genes are available in the same crop species, the transfer of disease resistant genes between the crops is done by sexual hybridization. For example, the yellow mosaic virus resistant variety of bindi, Bourboni grandi, is grown by the transfer of disease resistant genes from its wild relatives. Although many disease resistant hybrid varieties are produced through conventional breeding, the process is limited by the number of disease resistant genes available in different crop varieties and the wild relatives. As a result, another plant breeding technique called mutation breeding is widely employed. Mutation causes changes in the base sequence within genes, resulting in a new character not found in parental type and results in genetic variation. Mutation breeding involves the use of chemicals or gamma radiations to induce alterations in the genetic makeup of crops. The second generation of mutation induced crops possess new characters that are absent in parents. These plants that are screened for disease resistant genes and then artificially picked for breeding. An example of mutant breeding is seen in mung bean variety that is resistant to the yellow mosaic virus and powdery mildew. Apart from pathogens, pests also reduce crop production. As a result, there is a need to develop crops that are resistant to pests as well by using plant breeding methods. This involves the same steps that are involved in the development of disease resistant plants. Germplasm of cultivars or wild relatives are used as a source of pest resistant genes. Insect resistance in crop plants can be attributed to the morphological, biochemical, or physiological features of a crop. For example, hairy leaves on cotton make it resistant to jacids, while hairy leaves on wheat make it resistant to cereal leaf beetle. Also, smooth leaves and the absence of nectar in some cotton varieties protect them from bollworms. Similarly, solid stems of wheat protected from stems of fly, whereas maize is protected from stem borers due to its high aspartate and low nitrogen and sugar content. There are quite a few insect resistant hybrid varieties developed through plant breeding. Pusa Gaurav, a hybrid variety of rapeseed mustard, is resistant to aphids, while Pusa Sem 2 and Pusa Sem 3 are hybrid varieties of flat beans that are resistant to jacids, aphids, and fruit borers. And the hybrid varieties of Bindi, Pusa Savani, and Pusa A4 are resistant to shoot and fruit borers. In this manner, plant breeding techniques help improve crop production as well as prevent the use of bactericides and fungicides that can be harmful. They help develop crops that are resistant to pests and pathogens and can assist in boosting food production.